So the stuff you need, guys, obviously it's in Google Classroom. Okay, so for in there, you'll find it under, it's in the stream, okay, here as well. Okay, but it's probably easier to see it in the classwork tab. Right. So what you're going to need is this file over here. It'll say your name, and then it'll be like lab report expectations / format. Okay, that's the one you want to have open, because that's the one I'll be working out of for the next few minutes. And then we'll move over to, okay, the lab report file, which is what you're going to turn in for me, okay, when this is all done. All right. Okay, so for our lab report format and expectations, yesterday we talked about what's going to be in a lab report, problem, hypothesis, design, okay, all of that kind of stuff. Today we're going to go into detail about what's expected in each section. Okay, what am I specifically looking for from you in each section? Okay, so for the first part, yesterday we said, okay, there'll be a problem. I'll have a problem written out, but it won't be in the form of a question. So what you'll have to do is restate it in the form of a question. Okay, so an easy example could be, what is the effect of light on plant growth? Okay, so that's the problem I'm investigating. What is the effect of light on plant growth? Okay. In the design part, okay, you need to identify your variables. Every experiment has one manipulated variable, one or more responding variables, usually just one, but there's occasions where one of our labs may have two, okay, responding variables, and many control variables. Right? You will be required to list 3 control variables in every lab report. Right? So as a reminder, the manipulated variable is the thing that you as the experimenter are changing in the experiment. You are testing to see what effect it has on the responding variable. Okay? So in a lab where I'm trying to find out what the effect of light is on plant growth, which one of these 2 things is my manipulated variable? Light. I can change the amount of light a plant receives. In response to that, the plant will grow differently. Okay, so the growth of the plant is going to be the responding variable, right? And I may measure that in a number of ways. So there may actually be a number of responding variables in terms of how I'm going to measure the growth of the plant. Okay, but what we're going to be looking at, okay, is how do I tell the difference and how do I explain this? Okay, so in my design, my manipulated variable is the part of the experiment changed on purpose. Okay, so what I'm going to do in this experiment is likely have a few different plants, maybe 6 different plants, and they're all going to get different amounts of light. Okay, that way I can compare what the effect of light is on plant growth. Okay, so that would be how I'm manipulating the light. Okay, so it's changed on purpose by the experimenter. It's what's being tested. So the example here would be if I'm writing out my lab report, okay, I would say the manipulated variable is the amount of light given to plants because all subjects will receive different amounts of light and light powers photosynthesis. Okay, so I've identified that light, the amount of it specifically, is what my manipulated variable is, and I've explained how it's being manipulated and why. Okay, that's what needs to be in each and every explanation / identification of a variable. Okay? We need to say, here's the variable, here's how it's being manipulated, and why. Okay? If all you do is write light, you're not going to get full marks. Okay? You have to not only identify, but also explain. Everybody kind of following me there? Okay. Then I have to identify my responding variable, or variables in some cases. Okay? So here we're just going to be very general. Okay? The responding variable is whatever changes as a result of the manipulations made by the experimenter. So in this example, the growth of the plants is the responding variable because plants getting more light have more energy for growth. Okay? So I've identified the responding variable as the growth of the plants, and I've explained why it is the responding variable. It's responding to how much light is being given. Okay? It's going to change as a result of that. Okay, everyone follow me there. Okay? Another example could be if I was testing, let's say, an allergy medication. Okay? My manipulated variable would likely be the dosage of the allergy medication. Okay? My responding variable would be the relief of the symptoms. Okay? I would be watching for how people felt. Okay? Did their itchy eyes and runny nose and all of that stuff go away? Okay? Responding to how much of the medication they got. Right? Now, within that group, would I have to have some people who don't get any medication? Yes, because I need to know what happens. What if they just get better, okay, without getting anything? Right? I need to know that too. It's called the placebo effect. Okay? I would give them a sugar pill with no medication in it. That way they still take a pill. They don't know they're not getting the medication. They took a pill. Okay? It's what's called a blind test. Okay? And I would still have this responding variable, but obviously for that group, the people who don't get it, I should see no change in the responding variable. Okay? All the other people, I should see a change. Okay. Then we have our controlled variables. Now, controlled variables are usually the place where I can see how well you understand what we're doing. Okay? If I am testing the effect of light on plant growth, I need to make sure there's nothing else in the experiment that could affect the growth of the plant. In other words, I need to keep all the other things that affect the growth of a plant consistent or controlled between all of the plants in my experiment. If there are any differences, they could give me bad data. All right, so what I would need to do is understand what other things affect the growth of a plant. Well, the amount of water that they get, the soil that they're growing in, the temperature of the air they're growing in, all of that stuff can affect how they grow. So I need to make sure that that is kept the same. If I don't, and I give some of the plants more water than others, I'm not going to know whether it was the light or the water that caused them to grow differently. And then my experiment is meaningless. Okay? Everybody follow me there? Okay? So that's the importance of controlled variables. Control everything else that could affect the lab so that you don't get interference with your data. Okay? If I was doing that allergy medication thing, I would have to make sure that none of the people in my test are on any other medication. Okay? Otherwise, that could interfere with my test. Okay, so for this lab, okay, so the controlled variables would be, okay, so there are other factors that could affect the outcome of the experiment if left unchecked. So, same species of plant. I wouldn't want to compare an apple tree to a carrot to a bean. Okay? They all grow differently. It's not a fair experiment. All right? So, same species of plant because different plants grow at different rates. So I'm controlling that. They're all carrots. Okay? Same amount of water because water is an important factor in plant growth, so each plant needs the same amount so it can't affect the growth rate of the plants. All right, so I've stated I'm controlling this because I know it helps plants grow and I don't want it to interfere with the lab. Okay? Same soil would be another control variable. Plants get nutrients from the soil and different soils hold water differently. Keeping them the same ensures it cannot affect the experiment. Okay, and I've even listed a fourth one here, same temperature, as plants typically slow their growth in extreme heat or cold. Choosing an effective temperature for all plants ensures it does not affect the experiment. Okay, I'm keeping all these the same, and when I identify them, I explain the effect they could have and how I'm controlling them. Okay, those are the two things that need to be in your explanation of each controlled variable. Okay, here's how it would affect the experiment. Here's how it's being controlled. Everybody follow? Okay. Most often what I see here is people getting lazy. Okay? They'll say controlled variables, same species, same amount of water, same soil, same temperature, because it affects the plant. And they try and use that short explanation to cover all of their controlled variables. Each variable has a different effect and has to be controlled differently. Okay, so we need to explain each one on its own. All right, questions on the design part? Okay, so remember, that's 5 marks of the lab report. Identifying and explaining your variables. Okay, when you've done that, then you have your hypothesis. And we talked about the hypothesis yesterday. A hypothesis always contains these 3 words. If, and, then. Okay? It is a deductive statement about what you think is going on how you're going to test it, and predicted results. Okay? If this is true and this is done, then this will happen. Okay? That way, any good hypothesis, if it's written properly, can be absolutely rejected or absolutely accepted at the end of the experiment. You cannot sit on the fence with your hypothesis. You have to choose a side. Okay? If you're wrong, so be it. If you're right, great. Okay? But you have to choose a side. All right, so an if-then statement, if and then statement that communicates what the relationship between the manipulated and responding variable is thought to be. That's the if part. Okay? Briefly describes the experiment. That's the and part. Okay? And outlines the expected results. That's the then part. Okay? So those are the three parts. Okay? So an example could be, if plants require light for growth. So that's what I think the relationship is between light and plant growth. I think it's required. Okay? And 5 identical plants are given different amounts of light. So just a brief description. I didn't go into detail about the soil and the temperature and all of that. I just said this is a brief description of what I'm doing. Okay? Then the plants that receive the most light will show the most growth. Okay, am I going to be right here?
You don't know at this stage.
What's that?
You don't really know.
I don't really know at this stage if I'm actually doing this experiment, but what I do know is that you can get too much of a good thing. Plants don't want to get 24 hours of sunshine. Okay? They're not designed for that. It'll kill them. Okay? So I'm going to end up being wrong, and that's okay. Right? Okay, questions on how that works? Okay, so there's more detail here. If, what is the cause of the problem or the relationship being investigated, and briefly describes the experiment, then what to expect if you are correct. Okay, always goes in that way, right? Don't just say, plants with the most light will grow the most. That's not a hypothesis. Okay, that's like a guess. Okay, a hypothesis has to have the if, and then part. All right, then you will have your procedure. And for this lab, we're going to actually type that out ourselves, make it up ourselves, and I'm going to help you with that when we get to that point. Okay. Most of the time, though, it'll already be in the experiment. It'll already be in the lab report. Okay. You don't have to do anything with it except follow it in the lab. Okay. This will be the only lab where you actually have to write it out. Then you're going to have your observations. You have to decide how you want to record those. Is a chart the best idea? If it is, I'll probably have already built one and put it in the template lab report for you. Okay. If it's having to make a graph or just record anecdotal things, where you're writing out today the plants looked a little faded or whatever. Like you have to decide what's best for what I need to record and choose the appropriate way to do it. Okay. But it's your charts, your graphs, and any other data. Okay, including any notes. Okay, acquired during the experiment. Right, so that'll be out of 3 marks. Okay, basically I'm just looking to see if A, you have them there, and B, they're correct. Okay. All right, for the analysis, okay, the analysis is usually going to be questions to answer, okay, or calculations to perform. I might be looking for graphs and things like that. Okay, sometimes if you have to actually build them, right? But most of the time it's just going to be you answering questions. In this first lab, it's going to be whether you identify the compounds correctly. Okay? So it'll just be a chart that says A, B, C, D, E, okay, and then identity, and you just have to fill it in. Okay? The more you get right, the more out of 5 you'll get. Okay? So sometimes it'll look like that. Okay, in other cases, I may want detailed explanatory answers. If we're in the biology unit, we're talking about cell structure you saw under the microscope, I'm probably going to want you to describe more, talk more, write more. Okay? Last thing we'll have is our conclusion. So for your conclusion, you restate your hypothesis, which just copy and paste it. Okay, that's fine. Save yourself some time. Okay, then use your data and analysis to explain why your hypothesis is accepted or rejected. All right, so if I go back to my observations, let's say that in my experiment with the 5 plants, okay, I had one plant in the dark, 24 hours a day. Okay, I had one plant that got, let's say 6 hours of light, one that got 12 hours, one that got 18 hours, one that got 24 hours. That's 5 plants, right? Okay, so those are my plants. What I find is plant was in the dark, died. Plant that was in the light all the time, died. Okay? Plants in the middle did okay. Best plant, the 12-hour plant. Right? So, what that tells me, okay, what I get out of my analysis is that it doesn't seem like more light is necessarily good. Okay? To a point it's good, but there's a point where it's bad. Okay? That's what I get out of my analysis. All right? So then in my conclusion, I talk about that. And I say, the data shows that total darkness kills a plant. It also shows that total brightness kills a plant. Okay? This would seem to imply that plants require some periods of light where they receive energy and some periods of dark where they don't. Okay? And this seems to be, the optimal amount seems to be 12 hours of each. Okay? That's what I've written, or what I've discovered. Then I would write after that, therefore, the hypothesis is rejected. Okay? I've explained what my data and analysis say, and then I say, because of this, I cannot accept this hypothesis. Okay? It was not correct. I said more light would be better, and it's not. Therefore, I'm not right. Okay? Everybody follow on that? Okay? You can't just in the conclusion go, I was right. Good for you. Okay? You have to explain why. Okay? Here's what happened in the experiment, and here's how it relates to my hypothesis. Hypothesis is accepted, or hypothesis is rejected, one or the other. Okay? Will you lose marks if your hypothesis gets rejected? No. Okay? Lots of hypotheses get rejected. Most of them. Okay? That's how we learn things. Then you go, okay, well that didn't work, so I'm going to come up with a new hypothesis and try a different experiment. Okay? That's how it goes. Right? So that's what I'm looking for in the conclusion. So analysis is out of 5 marks, conclusion is out of 4 marks, and then the last thing is 2 sources of error and suggest improvements. All right? So for my lab here with the plants, okay, maybe I had my experiment set up so that I put plants in front of a window. But that meant that I had to keep coming in and out, okay, moving the plants around. Some days it was cloudy, some days it was sunny, okay. It was hard to regulate the temperature because of that, okay. I had the one plant that was in the dark all the time and all that kind of stuff. So I would say a source of error then would be that my plants didn't consistently get the same kind of light. Okay? I could not have given my plant that was supposed to get 24 hours of light, sunlight. It had to get artificial light for a lot of those hours. Is that the same? It's not. Okay? That's a source of error. Maybe the type of light interfered with my results. Okay? All right, so I would have to mention in my sources of error that I had to change the sources of lighting during the experiment, okay? And that may have affected my results. In the future, I would use artificial light for all of the plants. Okay? That way, at least it would be more controlled than what I did. Okay? Now, that's a source of error within the experiment, within the design, within the procedure that I made. Okay? It's not a mistake that I made and didn't correct. It was me following and then realizing that I probably could have done something better. Okay? Everybody with me on that? Okay. What we don't want to say in this section is I screwed up, I didn't fix it, okay? And as a result I got bad data and basically everything past this is meaningless. Okay? We don't want to say that. So for example, for our lab coming up, if you accidentally spill a little bit of sample A into the depression for sample B, and you just keep doing the experiment without changing anything, okay, and then at the end you go, yeah, source of error. I contaminated sample B with sample A. I went ahead and did all the results with the contaminated sample, so my data is probably not very good and I really can't be sure what this is. Okay? That's human error. You knew you did it. You knew how to fix it, and you did nothing. Okay? That's human error, not experimental error. Okay? And you don't want me to get that message. Okay? Because the message I receive if I read something like that is, hey, I'm super lazy and I just didn't want to fix it. Okay? You can't say that. That's not good science. Okay? So you only identify things that are within the experiment, okay, that are sources of error. Things you could do nothing about, but made your life difficult, okay, or made it possible that there could have been errors in your data, okay, but they were beyond your control. Okay? If it was just a screw up, fix it. Okay? But if it was something I was told to do it this way, but I found that it gave me trouble with this, that's a source of error. Okay, everybody follow the difference there. All right, that will be out of 3 marks. Okay, and your last 5 marks are just an overall. Okay, and that's, you know, things like, is it organized, is it thorough, is it full of spelling mistakes, full of grammar errors. Like, guys, if I open up your file and it's bleeding, okay, what I mean by that is there's so many little red things underlined, okay, because they're spelling mistakes or whatever, that doesn't give a good impression. Okay, so you got to make sure you run the spell checker, okay, all of that kind of stuff. Okay, otherwise it leaves a really bad impression. All right, questions on format and expectations? I'm going to give you about, let's say, a 5-minute break here, okay, and then I'm going to start into the actual lab report stuff, okay, and we can get started working on that today. Okay, so I'll give you 5 minutes here and then we'll do that.